It's a pleasure to talk to you today about the development of therapeutics for facioscapulohumeral muscular dystrophy. I have no relevant conflict of interest. So facioscapulohumeral muscular dystrophy, or FSHD, is one of the most common muscular dystrophies. It has a prevalence of 1 to 8,000 to 1 in 20,000. And it's an autosomal dominant disorder of families affecting mother and son. As you can see, have an inability to smile, which is a hallmark of the disease. It's an extremely disabling but not fatal disorder. Now, the genetics and epigenetics of FSHD are complex. In brief, 95% of FSHD, or FSHD1, is due to contractions of macrocytolite D4Z4 repeats in the 4Q subtelomeric region. This leads to hypomethylation, and in the appropriate context, to the misexpression of a double homeobox protein DUX4. Another 5% of FSHD, or FSHD2, is due to mutations in a gene SMCHD1, which leads to hypomethylation without contraction in this region, and also a misexpression of DUX4. It's possible that DUX4 itself, which has been shown to be toxic, leads to the myonecrosis, or that any of its downstream target genes leads to pathology. So various targets for FSHD therapeutics have been proposed. At the 4Q subtelomeric locus, one could overexpress SMCHD1 or modulate histone acetylation to repress DUX4. One could target DUX4 and knock down messenger RNA expression, or inhibit DUX4 protein or binding partners. One could target DUX4 target genes themselves if we knew which were the ones responsible for pathology. Whereas is currently ongoing in clinical trials, one could target downstream pathology with immunomodulatory drugs or anabolic agents to increase muscle growth. So the approach that I and others have taken is to try to silence DUX4 with antisense oligonucleotides. These are chemically modified single-stranded DNA and RNA, or analogs, targeting specific messenger RNA and pre-messenger RNA. And this shows the structure of the phosphodiamerate morpholino. This is a 6-membered morpholino ring, which substitutes for a ribose or deoxyribose. And antisense oligonucleotides are currently showing a lot of promise for other muscular dystrophies, Duchenne muscular dystrophies and myotonic muscular dystrophies. And others, including the DiMonso lab, have previously shown that targeting DUX4 messenger RNAs using antisense oligonucleotides efficiently silences DUX4 in vitro. So we similarly began our experimental approach with a screen of various different morpholinos to the DUX4 transcript. And morpholinos were produced for us by Genzyme. The specific sequences are in our Molecular Therapy 2016 paper. The ones that we took forward included FM10, directed at the splice acceptor site of exon 1 and 2, FM8, 9, and 10, to regions around the polyadenylation signal. In our screen, since we had previously shown that DUX4 is turned on with differentiation, we looked at FSHD myotubes that were cultured for 4 days in differentiation medium, and then immunostained for DUX4 full-length protein, or did RT-qPCR for DUX4 target genes. PMO effects of DUX4 protein were evaluated by control or DUX4 PMO treated myotubes, and with an antibody to the C-terminal region of the DUX4 protein. And we had a significant knockdown with FM10 of total DUX4 positive cells and DUX4 nuclei, and almost a complete abolishment of staining with FM10. Now, FM9 in green, and to a greater extent FM10 in purple, knocked down DUX4 target genes, showing that these morpholinos interfere with DUX4 function. Importantly, myosin heavy chain, a mark of myogenic differentiation, was not altered by these morpholinos. The FM10's ability to knock down DUX4 target genes was dose-dependent, with an ED50 of 1 to 3 micromolars and a maximum effect at 10 to 20 micromolars without any signs of cell toxicity. So to develop a reference transcriptional signature of FSHD, we did RNAseq on human myotubes from FSHD subjects and unaffected subjects. And these are volcano plots showing you the log 2-fold change versus the negative 10 p-values. And genes which were transcribed higher in FSHD are in red. And this provided us with a transcriptional signature of FSHD. And we found that many of these genes, similar to others have reported, were DUX4 target genes shown in black boxes. Now in FSHD myotubes, if we look at those treated with FM10 versus control, we find that what the major effect is a downregulation of the transcriptional signature. That is, that those genes that were upregulated in FSHD are downregulated with FM10 and colored in red. If we look at the unaffected myotubes, this gives us a window into any potential off-target effects. Unaffected myotubes have little, if any, DUX4 expression. So in FM10 versus control, treated unaffected myotubes, we find only 3 genes were differentially expressed, which is approximately what one would get by chance from assaying 37,000 genes. And so therefore, FM10 did not have any significant off-target effects in these myotubes. So we wanted to determine whether our lead compound, FM10, could knock down DUX4 in vivo as it has in vitro. And there are several different models of FSHD in vivo. We wanted a model in which DUX4 was just lowly expressed, as it is in humans, and one that might be able to predict a clinical response in the future. And so for this reason, we constructed xenografts of FSHD, in which we took muscle from healthy donors and from FSHD donors and transplanted into immunodeficient mice. So the muscle that is transplanted and cut, the myofibers degenerate, but the satellite cells in the muscles become activated, differentiate, and form new muscle in the mouse, which is innervated by the mouse motor neuron. This is what the tissue looks at like 2 weeks after transplantation. You can see that there still is some degeneration, but there's areas of regeneration, whorls of human fibers marked by human spectrin within circles of human collagen, suggesting regeneration is occurring within the existing basal lamina. And at several weeks post-transplantation, you can see that the muscle is fully regenerated and is almost completely human in origin, as shown by anti-human spectrin antibodies. Co-labeling with human laminin C, PAX7, which is a marker of satellite cells, human laminin, we can identify several different cell types, including interstitial cells, myonuclei, and satellite cells. And all of these human nuclei populations are on the order of 90% human within the graft. The vasculature within the graft is a combination of human in green and mouse in red, and there are areas, such as here, where it appears that the human vasculature and the mouse vasculature are anastomose. And indeed, we find mouse red blood cells within the human capillaries, indicating that the human capillaries are patent and functional. So we've established that the xenograft is a feasible model of human muscle with predominantly human myofibers and human nuclei. We then wanted to determine whether it was a functionally competent muscle. We identified neuromuscular junctions by co-labeling with presynaptic and postsynaptic proteins, and the presence of type 1 fibers in purple and type 2 fibers in green indicate that more than one motor neuron innervates the xenograft. We then looked at the function of the xenografts by assaying contractility to trains of action potentials at increasing frequencies. At low frequency, you can see a twitch response, and at increasing frequency, there's a summation to tetanic force. The xenografts exhibit a steep force-frequency relationship, which is consistent with functionally competent muscle. We can also look at the function of the xenograft by looking at excitation-contraction coupling. For these experiments, we enzymatically digest the xenograft and look at individual myofibers, which we confirm are human with immunostaining. We load the myofibers with a calcium indicator dye, Indo-1, and subject to electrical field pulses. And the robust calcium release that you see from single pulses or tetanic trains of pulses indicates that the excitation-contraction coupling system is intact. Now, important for a model of FSHD, these xenografts from donors who had FSHD expressed DUX4, while xenografts from control donors do not express DUX4, similar to the biopsies from which they originated. And the general gene expression profile of multiple xenografts from multiple donors also mirrors the donors from which they originated. So here I've shown the differential expression of FSHD versus control for biopsies versus the differential expression for xenografts. And the dotted black line is what would be a complete agreement between the expression in xenografts versus biopsies. And the dotted red line is the best fit of the data by orthogonal regression. So I hope that I've convinced you that the xenograft is a valid model of FSHD. We then took this model to determine whether FM10 could knock down DUX4 in vivo. We used for these experiments xenografts that were 6 months old. We treated with standard morpholino or FM10 morpholino by electrophoresis, and then after 2 weeks analyzed the muscle. We first determined that the muscle was fully human by antispectrin and lamin AC. And we looked at genes including DUX4 full length and DUX4 target genes, MBD3L5 and ZSCAN4 in comparison to housekeeping genes. And we looked at xenografts which came from living donors as well as ones that came from an autopsy and treated with standard morpholino or FM10 morpholino. And these are just the numbers of xenografts used in these experiments. And this is my last data slide, which is the culmination of these experiments, showing that FM10 dramatically and consistently knocked down DUX4 expression, whether the xenografts came from living biopsies or from autopsies. In addition, the FM10 knocked down MBD3L5 and ZSCAN4 very consistently, suggesting that it interfered with DUX4 function. So I'll conclude then that FSHD can be modeled in vivo by xenografting human muscle into immunodeficient mice. PMO is directed at the polyadenylation signal of DUX4 consistently and profoundly knock down DUX4 and DUX4 target genes in vitro and in vivo. And therefore, we believe that antisense oligonucleotide targeting in DUX4 is a promising therapeutic approach for FSHD. I'd like to acknowledge Tracy Jang, who did much of she's a graduate student who did much of the work in my lab. Jennifer Chen, Charlie Emerson, Oliver King, Fedik Ramov, and Lou Kunkel, all from the UMass Wellstone Center for FSHD. Chris Ward at the University of Maryland, Nick Clayton and Bruce Wentworth, who was then at Genzyme, Terry Partridge at the National Children's Medical Center. And I'll shamelessly put in a plug that if anyone in the audience is looking for a postdoc and found this work interesting, please come talk to me. Thank you.
Catherine, I'd like to ask a question, a couple questions. One, what do you see that might be the role of the immune system that we don't have present in this model? and how might that contribute to the effectiveness of a therapy?
So it's really unclear how much FSHD is an inflammatory or immune-mediated disorder. The company aTyr is certainly banking on that with its compound directed to immunomodulation of FSHD. We can see edema or increased fluid content in muscles on MRI that is suggestive of inflammation in FSHD. And this model does not have inflammation, so we wouldn't be able to assay those effects. What we can say is that the molecular mechanisms, knockdown of DUX4 is accomplished in this model.
And my second question is, there might be some trickiness in applying therapies because we know that DUX4 is probably a very focal and quick expression. Do you think that might play a role in how we really approach this disease?
Yeah, it's certainly a concern. The DUX4 is expressed in incredibly low levels, maybe 1 in 10,000 nuclei express it, maybe less than that from your work. If we have a drug that is trying to catch those rare random events, it may be difficult. It's even possible that some of this is happening prenatally or in development. So that's a potential limitation of anti-DUX4 anti-nucleotides.
Thank you very much. It was a great talk. Sure.
That was great. Can I ask you a question? So with the Xenograft being efficiently supplied by Vasculature, so your model would also be, it would be feasible to test intravascular delivery of these morpholinos?
Yes.
Or systemic delivery?
Yeah. We did, actually. We tried some peptide-conjugated morpholinos that were too toxic, so we're currently in discussions with Sarepta to develop novel peptide-conjugated morpholinos.
Very nice talk. I have two questions. One is how long the knockdown lasted?
So we only looked at the 2-week time point.
So do you have any plan to follow up to see how long it will last and if you need to do repeat treatment?
Yeah, I think that we will need to look at the half-life of DUX4 expression.
And also, have you looked at the off-target effect?
The off-target effect, from the RNAseq looks like it's minimal.
Thank you.
Mm-hmm.
How long does the Xenograft actually last?
So the Xenograft lasts for the life of the animal. So we've looked at it over a year out. One of the negatives to the Xenograft is that it takes a very long time to mature. We didn't actually need to use 6-month-old animals. Those are the animals that we had on hand. But it does take a couple months to mature, and I believe that's from the NRG model not having immunological cells to really stimulate the regeneration fully. Thank you.
Thanks.